As we know, Salmonella is one of the major causes of foodborne disease illness worldwide and can contaminate various food products such as poultry, eggs, meat, and dairy products. Consumption of food contaminated with Salmonella may cause symptoms such as diarrhea, fever, abdominal cramps, and vomiting. Therefore, the detection of Salmonella in food samples is important to ensure food safety and public health. In this experiment, the conventional method based on the FDA Bacteriological Analytical Manual (BAM) was used to detect the presence of Salmonella in food samples. This method involves several important steps including pre-enrichment, selective enrichment, isolation on selective agar media, and biochemical identification. Different selective media are used to encourage the growth of Salmonella while inhibiting other microorganisms. The purpose of this experiment is to isolate and identify Salmonella from food samples using conventional microbiological techniques. Through this practical, students are able to understand the principles of food microbiology, bacteria isolation, and biochemical testing used in the detection of foodborne pathogens.
Assalamualaikum warahmatullahi wabarakatuh. So my name is Alif Ekal, number matric S76973. So today I'm going to talk about the objective of this experiment. So the objectives of the experiment is to detect the presence of Salmonella species in the food sample using microbiological method. Next is to learn and perform the procedure involved in the isolation and identification of Salmonella for contaminated food sample. The third one is to understand the importance of selective media and biochemical tests in the detection of foodborne pathogen like Salmonella. And the final one is to evaluate the microbiological safety and quality of food sample based on the presence of or absence of the Salmonella in the. So as you can see, the xylylysine dissolute chocolate agar both on the RVS and TT have a strong black colony growth. Next is for the bichromous sulfide agar, we got no black colony growth. So next is for the RVS on BS agar, so no blackening showing on the RVS on BS agar. Next on the TT on XLD agar, we get a strong black colony growth. Next is for BS on TT, we have a strong black colony growth, while for the XLD on RVS, we also have a strong black colony growth.
The objective of this practical session was to detect the presumptive presence of Salmonella spp in a raw meat sample utilizing selective enrichment followed by indifferential isolation plating. Raw meat products are highly vulnerable matrices frequently implicated as reservoirs for Salmonella contamination, demanding rigorous monitoring in food safety frameworks. Following standard enrichment phases designed to suppress background microbiota while allowing sublethally injured pathogen cells to proliferate. The sample was streaked onto two highly selective media, which is bismuth sulfide agar (BSA) and xylose lysine deoxolate (XLD agar). The experimental outcomes yielded highly distinct positive presumptive results on both selective medias. On the BSA plate, the development of characteristic distinct black or grey dark colonies accompanied by a dark precipitate in the surrounding medium and distinctive metallic sheen strongly that implies the presence of Salmonella. The unique selectivity of BSA is achieved through the bismuth sulfide and brilliant green dye, which works synergistically to inhibit the gram-positive bacteria and competing coliforms. Salmonella species successfully proliferate and reduce the sulfide indicators into hydrogen sulfide gas. This gas then binds with ferrous sulfate embedded in the formulation to form an insoluble black ion precipitate, altering the color of both the colony and the surrounding agar. The phenotypic identification was conclusively corroborated by the results observed on the XLD agar, which demonstrated red translucent colonies featuring distinct black centers. On XLD, Salmonella metabolized xylose and rapidly exhausted it, triggering the alkaline decarbolization of lysine, which reverts the phenol red indicator back to a deep red hue. Simultaneously, hydrogen gas H2S production manifests as a prominent black centers due to its reaction with the ferric ammonium citrate. Together, these mutually supportively biochemical profiles provided by the robust presumptive confirmation of Salmonella spp. Contamination in the meat sample underscoring the urgent necessity for strict sanitization controls and critical control point monitoring across the supply chain.